Hello, my name is Bradley Walker, and today I will be presenting my essential nutrient vitamin B6, or pyridoxine. First, let's get into some detail about the structure of this molecule. Vitamin B6 consists of a family of 6 variants of an anomeric carbon-based molecule. Substituted carbon groups are depicted here, where you have an alcohol group over here on this one, an amine group on this one, and a carbonyl group coming off of this one. All 3 of these forms can be interconverted in the body, and all of them can be phosphorylated to increase the variety of their function. So, some sources of vitamin B6 that are common in your diet include organ meats, such as liver, brain, and intestinal tissue, potatoes and other starchy vegetables, and fruits, excluding citruses, so apples, pears, and bananas. The NIH reports grain-based foods and cereals as a good source of vitamin B6, along with poultry, such as chicken and turkey, and fin fish, such as tuna and trout. The NIH also reports some recommended intakes for adults, ranging from 19 to 50 years old. So the recommended intake per day is 1.6 milligrams of vitamin B6. Now this number varies in the case of pregnancy or during lactation, where more is required to meet the needs of these two conditions. The maximum recommended intake of vitamin B6 for individuals above the age of 19 is 100 milligrams per day. The functions of B6 include the regulation of metabolic enzymes, where it acts as a cofactor for over 100 different enzymes. Important reactions it is involved in include the synthesis of heme groups for hemoglobin and myoglobin, as well as reactions that break down amino acids. It also functions in neurological development, as it aids in the synthesis of important neurotransmitters like serotonin, dopamine, and gamma-aminobutyric acid, or GABA. Vitamin B6's role in the immune system involves the assistance with the absorption of B12 into the blood, which then creates red blood cells and T cells important in immune responses. For this presentation, I will focus on the role of B6 in metabolic regulation of amino acid catabolism, specifically the catabolism of homocysteine. Metabolism of amino acids takes place in the stomach and small intestines. In the stomach, pepsinogen is secreted, which is then converted to pepsin to break down proteins in the stomach. Trypsin, chymotrypsin, and elastase further break down proteins in the small intestines. Enzyme cofactors are important molecules that control the activity of zymogens, or inactive enzymes, such as pepsinogen in the stomach, and once they are activated, they then regulate the reaction that they were meant to regulate, such as, in this case, protein digestion. The amino acids produced by protein catabolism are important for structural reasons, for cell signaling, for membrane protein transport, and, of course, can also be broken down to produce energy when glucose levels are low in a starvation state. Homocysteine is an amino acid resembling cysteine with the addition of a methyl group. This methyl group is important in its role as the methyl donor in the methylation of methionine, another amino acid. This methylation process is dependent on 2 B vitamins, B12 and B6. We'll focus on B6 in this instance as it plays a crucial role in the conversion of homocysteine to the less harmful cysteine amino acid. So here you can see the 2 reactions that vitamin B6 help regulate as a cofactor. So homocysteine is first converted to cystathionine with the help of vitamin B6 by cystathionine B synthase. Cystathionine is then converted directly into cysteine with the help of vitamin B6 via cystathionase. So this reaction it converts homocysteine all the way to cysteine with the help of vitamin B6. So, the importance of this conversion, I will go into detail about the effect of homocysteine and the reason that vitamin B6 deficiency can lead to cardiovascular diseases. So, high concentration of homocysteine can lead to a condition called hypercystinemia. This condition causes toxic effects to endothelial cells, promoting proliferation, formation of lipoproteins in the blood, and the activation of procoagulant molecules. The collection of these things leads to a decrease in the diameter of blood vessels, eventually leading to cardiovascular diseases, such as atherosclerosis, heart attacks, and strokes. Since B6 plays a role in reducing the concentration of homocysteine as it converts it into cysteine, it is an important factor in reducing the risk of CVDs. The case study I chose explores the correlation between homocysteine and vitamin B6 in the hopes that it can find a way to medicate the increased concentration of homocysteines in patients with schizophrenia. For this study, 11 individuals with schizophrenia were chosen, 7 males and 4 females from the ages of 28 to 66, presenting with a history of schizophrenia. They were given B6 treatments over the course of 12 weeks. Blood samples were obtained at baseline and after the 12-week supplementation period to assess the correlation of B6 vitamin concentrations versus the concentration of homocysteine. So the results after 12 weeks indicated a strong correlation between homocysteine concentrations and age, where older individuals had higher concentrations. This may be due to the overall lowered metabolic rate as age increases. A significant negative correlation in men was observed with a decrease in the concentration of homocysteine after the 12-week treatment period. This indicates that the supplement of B6 vitamins could potentially reduce homocysteine concentrations in men with schizophrenia. The lack of significant data for female patients prompts the need for further study at larger scales. The results of this study are a significant step in the direction of being able to clinically treat schizophrenia hyperhomocystinemia patients via vitamin B supplementation. Here are some references.